My presentation is about advancements in RNA molecule structure prediction using this thing called an SE3 equivariant transformer model, which I'll go into depth more later. And I conducted this research under my mentor, Maria Conte, who is a PhD from Georgia Tech. Okay. For some reason, okay. Understanding RNA prediction. So, RNA structure prediction is a critical area in computational biology that involves determining the actual three-dimensional shape of RNA molecules from their nucleotide sequences. Accurate predictions not only enhance our understanding of RNA functions, but also have far-reaching implications in drug development and disease diagnosis, which makes this field essential for advancements in biotechnology and medicine. Research questions, the importance of RNA structure prediction. Okay, understanding biological functions. So the ability to accurately predict three-dimensional structure of RNA is crucial for understanding its biological functions and roles. Basically, by knowing how RNA folds into an actual three-dimensional structure from its one-dimensional sequence of nucleotide bases, we can essentially predict its entire function. And that's very important for understandings of computational biology and the implications for drug development. So accurate RNA structure predictions have significant implications for drug development. By modeling RNA accurately, scientists can identify potential drug targets and design molecules that interact effectively with RNA. Challenges in RNA structure prediction. The challenge of predicting RNA structures stems from the vast number of possible conformations and the limited experimental data available for training models. This complexity necessitates innovative computational approaches to improve prediction accuracy. So the AI model that I used for RNA structure prediction leverages this thing called SE3 equivariance. This model is designed to predict the three-dimensional structure of RNA molecules by employing SE3 equivariance, which enables, so SE3 equivariance is basically a topic in like math, and what it is the symmetry in 3 dimensions, and applying it with this specific AI model allows it to understand all the rotations or translations of a 3D object, essentially eliminating a lot of error and improving the AI model's ability to generate precise predictions. Okay, so RNA is a messenger and the significance of RNA structure. RNA acts as a crucial messenger in the cell, transmitting genetic information from DNA to proteins. It regulates various biological processes, including gene expression and catalysis, which highlights its essential role in cellular function and organismal development. And then the significance of RNA structure. The three-dimensional conformation of RNA molecules is fundamental to their biological functions. Accurate prediction of RNA structure aids in drug development, disease modeling, therapeutic interventions. A deep understanding of RNA folding mechanisms is imperative for innovative medical solutions. Data sources and pre-processing steps. So this is how I actually went about training and testing the model. So I got a data set from Kaggle, which is a popular website with public data sets. And the primary data source is this Kaggle competition data set, which includes RNA sequences and their corresponding three-dimensional coordinates. Data normalization. Data normalization is performed to ensure that all features are on a similar scale, facilitating better training of the model. Data cleaning. So this one was very important in training the model. Cleaning the data set involves removing incomplete or inconsistent data to improve the quality of training data. When I was actually conducting my experiments, there was a lot of cases where there was data that was incomplete, so I had to just remove it or else it would interrupt the model's learning and make it less accurate. Data splitting. So the data is split into training, testing, and validation sets to effectively evaluate the model performance. And then the architecture of the RNA prediction model. So first we have the SE3 equivariant design, which is using that mathematical concept of SE3 equivariance, which allows it to understand rotations and translations during prediction. Then we have the multiple layers of this model. So this architecture is using multiple interaction layers to improve and enhance the model's learning process, which enables it to capture the intricate relationships between parts of RNA molecules and understand how it forms into a 3D structure. Enhanced prediction accuracy. By integrating geometric understanding, the model can better predict RNA structures, leading to improved accuracy in determining the 3D configurations based on RNA sequences. This approach is a significant advancement in computational biology. And performance and evaluation metrics. So there's two main ways that I evaluated the model, which is TM score, which measures the structural similarity, and then RMSD, that stands for root mean square distance, which is quantifying the coordinate divergence, basically figuring out the difference in distance from the actual 3D model from my predicted 3D model. And then enhancing RNA structure prediction through SE3 equivariance. So our proposed AI model will undergo rigorous comparison with current RNA structure prediction models because there's other ones out there. This evaluation aims to demonstrate the benefits of this SE3 equivariance, which I haven't seen widely used in other models, particularly in terms of enhanced prediction and addressing challenges that other models may face, such as not being able to understand the geometry of these molecules. So limitations in future work. There's a large dependency on training data. The current model's performance heavily relies on the quality and diversity of the training data. Inadequate or biased data sets can lead to suboptimal predictions and limit the model's generalizability to unseen RNA sequences. The limitations of computational resources. So I probably was not able to conduct my experiments as to the level of degree that's possible because I'm limited by my hardware. And then exploration of additional data sets. Future enhancements on this will focus on integrating more diverse data sets, which could improve the model's learning and accuracy. Additionally, the investigation of varying architectures will be pursued to optimize prediction capabilities and refinement of the model architecture. So changing and improving the model architecture is always possible. to give it better predictions. And this includes adding new layers, adjusting hyperparameters, and incorporating advanced techniques for better performance in prediction. Okay, so I believe this was a significant advancement in RNA structure prediction, introducing this SE3 equivariance to enhance prediction accuracy, because I haven't seen this used in other models that are trying to do RNA prediction. This improvement was essential for advancing applications in drug development and understanding genetic functions, ultimately contributing to ongoing research in computational biology. References and further reading. There's key papers on RNA prediction that you can find. You can research AI methodologies in biology. There's other data sets for RNA modeling. There is pretty recent advancements in RNA research that I've seen. There's new papers coming out pretty recently, and future directions in RNA studies. Okay, thank you for listening, and that's the end of my presentation.
Well done, Alex. Thank you so much for sharing this good work with us. We have time for just one or two brief questions. The first question I am personally curious about is that you mentioned you haven't seen this model, this work that you've done, widely used. So how might you advise a future researcher to incorporate this good work?
Yeah, I love that question. So, what I've seen in some of these papers is there was this large project, I forgot exactly what it was called, but they take a lot of models that are focused on RNA structure prediction, and they kind of have this way of combining all of them. So what I would advise to someone that's looking to incorporate my technique is doing something similar to that, where they maybe use multiple AI models to evaluate the same data and then combine their different predictions.
That's great feedback. Thank you. And then I guess a similar question is, earlier in your presentation, you mentioned the use of Kaggle, which is that publicly available data set. How might someone access this if we're endeavoring to make research in general more accessible? So walk us through how someone might work into that.
Yeah. So Kaggle is just a website. If you search up kaggle.com, I think, then they have a ton of publicly available data sets that you can use for your own AI research. And the specific data set that I used was called like the Stanford RNA prediction competition, I believe. And that was the data set that I used for this project. But yeah, Kaggle's a great website.
That's great, Alex. Thank you for teaching us about Kaggle and how to employ your research in the future. Well done. Thank you so very much.
Thank you.